next speaker has spent most of her academic life understanding a rather small but scary creature. And she has somehow tamed it for the sake of science. Pascale Cossart is emeritus professor at the Institut Pasteur in Paris. Her main focus of study has been Listeria monocytogenes, which is one of the most virulent foodborne pathogens we know. Her discoveries have illuminated how these bacteria invade and interact with host cells and how it manages to cross most of the barriers inside our bodies, including the one that protects our brain. Cossart has also turned Listeria into a model organism to expand our knowledge on bacterial infections. A member of multiple academies of sciences, she has received the L'Oréal UNESCO Award for Women in Science and the Robert Koch Prize. Please welcome Professor Cossart, who will present how we raise the bacterium to the rank of a model system, the Listeria paradigm.
Thank you. Thank you to the organizer for inviting me. This is a great honor, and it's a great honor for our discipline. And I would like to honor my discipline today by showing you how my lab has highlighted how a bacterium can produce an infectious process. So the organism that we are working on is Listeria. And since many of you don't know what is Listeria, I would like just to tell you that it's a bacterium which lives in the environment and can infect humans and some animals. So when we started to work on Listeria, not many things were known. It was known that Listeria is inducing a T-cell response and that antibodies are not responsible for the curing and the recovery from infection. I thought it was a fascinating infectious process because the bacteria, through contaminated food, arrive at the intestinal barrier. I'm going to try to manage this thing. Yes. So the bacteria arrive at the intestinal barrier and they can reach the brain and the placenta. This bacterium grows fast in both. It's not too pathogenic. It can infect tissue culture cells. It's intracellular. And there was an animal model. In addition, we knew two phenotypes. On blood agar plates, there was this halo. On egg yolk agar plates, there was this halo also. So, as you see from this slide, we have used a series of approaches. And I would like to highlight 4 approaches. We have used what we call cellular microbiology, which is a combination of genetic approaches coupled to cell biology and molecular biology. We use post-genomic approaches, hypothesis-driven approaches, and serendipity. So cellular microbiology is a discipline that we coined. We coined the name after a meeting that I co-organized by the 3 individuals who are here, and which really has the advantage to provide information on both the infectious process, the infectious agent, and the cells which is infected. So in fact, the launcher of that discipline, I give credit to Stanley Falco, who together with Ralph Isberg and many other people really launched the field of the interaction of bacteria with epithelial cells. So he was kind enough to cite Jorge Galland, Philip Sansonetti, and myself among the launcher of the discipline, which is now the title of departments, of reviews and chairs, etc. Okay, so the first thing that we did, there was nothing known about genetics, no gene were known. So the first thing that we did was to isolate a non-hemolytic mutant. This non-hemolytic mutant was avirulent, could not grow inside cells, and bacteria were absolutely avirulent. We clone sequenced the gene, and it was the first virulence factor identified in Listeria. The protein which is encoded by the gene allows the bacterium to escape from a vacuole, which is formed when the bacteria enter into cells. We had competitors at the time. They became friends, Dan Portnoy and Werner Goebel. So at that time, Dan Portnoy has discovered this very nice actin-based motility, and this scheme is absolutely classic now. He started to work on that after the Listeriolysin O competition field. So we were not interested to work on that at the time. And Werner Goebel was working on the regulatory factors regulating all the virulence of Listeria. We decided that we would work, sorry. It's blocked there. It's blocked. No. Okay, we decided to work on the entry of bacteria into cells. As you can see from this movie, Listeria has the capacity to enter into cells which are normally non-phagocytic. And we wonder what were the factors which are involved, both on the bacterial side and on the cellular side. So we identified a protein which is called internalin, which is allowing the bacteria to enter into cells. We identified the second protein that we call ENLB. And next to that, of course, we wanted to identify the receptor for this internalin protein. So we identify, after a chromatography affinity approach, a protein which is called E-cadherin. So for those of you who don't know what is E-cadherin, this is the yellow labeling that you see between epithelial cells. These proteins maintains the adherence between the two junctions, between the two cells. So on the left of this, I show you that instead of having homotypic interaction between E-cadherin and E-cadherin, in the case of Listeria, you have this internalin protein interacting with E-cadherin, and this allows the entry of Listeria. So we were, of course, interested in deciphering what, we were interested to decipher how the mechanism is acting, and we did a lot of cell biology to investigate what are the signaling pathways which are inducing this entry into the process. We had used human cells to identify E-cadherin, and we found that there was a problem because E-cadherin from human and E-cadherin from mouse are very, very similar, and Listeria does not enter into mouse cells. Not obvious. Maybe I can do that. Yes. So it was the work of Marc Lecuy to identify that there was a single amino acid difference in E-cadherin between human and mouse, which would confer the specificity. And after a lot of mutagenesis, etc., he was able to show that he could show that if you change this amino acid from mouse into human, you restore the entry into human cells. So he decided to generate transgenic mice, which would express only in the intestine human E-cadherin. And he would infect these mice by the oral route. And as you can see on the left, if you infect these mice, either with the wild type Listeria or the internalin mutant, there is no difference. The mice are perfectly happy. If you do that with the transgenic mice, which express the human E-cadherin in the intestine, you see that if you compare the wild type and the mutant, there is a difference of 100%. So this was a real proof that human E-cadherin expressed in the intestine allows the entry of the bacterium into the mice. Parenthesis, the immunologists were using the Listeria as a model by injecting the bacteria in the tail of the mice. In fact, in nature, mice are totally not susceptible to Listeria. So now we have a good model to study human listeriosis. So this was, okay. We have done a lot of experiments to investigate how the infectious process work. There is one experiment that I really like, is to show that, in fact, in contrast to what was written in the textbook, like here, this is not really working very well. Okay, sorry. I'm sorry. Okay, I'm not going to use that anymore. Okay, in contrast to what is written in the textbook, where you see that for the entry of bacteria, you have the role of actin, which is predominant. For the entry of small object, you have the clathrin, which is mediating the entry. And here we show that, in fact, the clathrin is mediating the entry of Listeria into cells. So we were going against the dogma, and we showed that actin and clathrin are absolutely critical for the entry of Listeria into cells. So that was it for the moment. We have identified the virulence factor Listeriolysine O. We have identified internalin for the entry. And now I would like to tell you what we found by studying the second phenotype that we had on egg yolk agar plate. we we searched for mutants, and we could identify a mutant which was not producing that, showing that it was affecting the lecithinase production of this bacterium. In fact, when Christine Cox arrived in my lab, I asked her to look at the behavior of this mutant into cells, and what she could show is that, in contrast to the wild type, which produces this magnificent actin tail inside cells, labeled in green here, the mutant was not producing that. And in fact, to make a long story short, we had identified a gene which was critical for this actin-based motility. We named this gene the ACT gene. So following that, the group of Tim Mitchison was able to identify the complex ARP2/3 complex, which binds to ACT and allows the actin-based motility. So what I find remarkable is that when you have a moving cell, like on the right of this slide, which is moving using the actin-based motility, there is a complex at the leading edge, which is made of ARP2/3 binding to SCA-WASP family protein. At the back end of the bacterium, the ARP2/3 complex is mediating the motility via interaction with ACT. And we have shown that ACT and WASP SCA family proteins are similar at the level of the amino acid composition. So it's a very nice example of some kind of similarity between mammalian cells and bacterial cells. Okay, so Listeria is not the only bacterium which is able to do that. It was shown by others that Shigella is able to move inside cells by recruiting the ARP2/3 complex via ICSA, which binds to NWASP. We have been interested in Rickettsia, and we found a protein which is binding to the ARP2/3 complex. Okay, so that was the Rickey protein. And we also were interested in the vaccinia virus, and we found that vaccinia virus is also mediating its motility via an actin-based movement, but we have not continued to work on these two projects. So I come back to the scheme that I have already presented, where you see that the bacteria are entering into cells. They escape from a vacuole via this Listeria O protein, LLO, and then they move inside cells, and they move from cell to cells via this ACT protein. At this stage, I would like to say that when we identified the ACT protein, we had a lecithinase mutant because ACT was interrupted in the mutant by a transposon, which had the polar effect on the PLCB gene, and that's why we had the lecithinase phenotype. Okay, so all the genes which were on the previous slides are regulated by a protein that we name PRFA, and this protein PRFA is a very interesting protein. It's only expressed at high temperature and positively regulates all these virulence genes. So when Jörgen Johansson arrived in my lab, I asked him to investigate more about this regulation, and what he found is very interesting, is that this PRFA protein is expressed at high temperature because it's regulated at low temperature by what we have called a thermosensor. So the gene, in fact, the gene is preceded by a stem and loop structure, which prevents the access of the ribosome when the stem and loop is formed. But when you raise the temperature, there is opening of the stem and loop structure. The ribosome can translate the message, and then there is virulence gene expression, which are co-regulated by this PRFA protein. And now there are other examples of thermosensors which are regulating virulence genes in other bacteria. We started at that stage to be highly interested in RNA, and we had in parallel been coordinating the sequence of the genome of Listeria monocytogenes and that of Listeria innocua, which is not pathogenic. And we wanted to extract from the comparison of these two genomes virulence factors which would contribute to the virulence of Listeria. And we found several. I'm going to give you five examples. We found that there were genes other than the one I had previously shown, which are regulated by PRFA. First, BSH, and second, ENLC. BSH means bile salt hydrolase. We found that Listeria encodes a bile salt hydrolase, and we could show that by constructing a mutant, we could show that, in fact, the bile salt hydrolase is used in the intestine to maintain Listeria when Listeria arrive in the intestine. So if you compare the wild type and the mutant in the feces of an animal which has been infected orally, you see that the persistence is dramatically affected. First results of our sequencing. Then we could find that ENLC is a protein which is secreted when Listeria is inside cells, and we could show that ENLC interacts with a subunit of the IKK kinase and prevent the normal expression of the immune response. I don't have time to go into details, just believe me, but this is just to illustrate that bacteria, upon infection, have many, many tools to really interfere with the many components that they meet in the cells. So LNTA is also a protein which is present in monocytogenes, absent in Listeria innocua, and it is secreted by the bacterium, and the protein goes to the nucleus. So we were able to show that it goes to the nucleus, and we started to call this family nucleomodulins. Well, so it goes to the nucleus, and what does it do? So we used the 2 hybrid screen and found a protein which was unknown. So we searched for the role of this protein, BHD1. We could show that this BHD1 is a protein which induces heterochromatin formation. But when the Listeria protein, LNTA, is produced, it desequesters this BHD1 and allows the secretion of proteins which are induced by the type 3 interferon. So this is another way that Listeria is interfering with the innate immune response. Okay, my fourth example is ZEA, which is a secreted RNA binding protein. This is the first bacterial protein which binds RNA, which is secreted. So this is the work of Alessandro Pagliuso, and he could show that Listeria is really secreting a protein which is always found with RNA. And we think it's in vesicles, but he's now investigating that in his own lab. So last but not the least, Nathalie Rollion found that there was a protein produced in monocytogenes absent in innocua, which is in the gut interacting with one single bacterium, which is Prevotella copri. And at this stage, we are trying to decipher what is the role of this inhibition of the growth of Prevotella copri. So this was not the first bacteriocin which had been investigated. In my lab, Javier Pizarro had been interested to decipher what was a protein named LLS, which had been identified in epidemic strains of Listeria, and he had found that this was a bacteriocin, which was really the key for this epidemic. So I think now Listeria has entered the field of microbiomes. Okay, after proteins, now we go to transcriptome. We were interested in the transcriptome of Listeria, and in fact, I was really interested to decipher whether there would be small RNA which would be involved in virulence. This is not exactly on what we have been focusing. I must say that at that time, we started a collaboration with Rotem Sorek, and together we investigated this old transcriptome. The first thing that we found was this, what I called excludon. This is not very well known for the moment, but I just would like to highlight the picture which is at the center of this slide, where you see that you have two operands which are divergent. And we found several examples of this situation where one operand can be expressed from one promoter or another. And depending on the environments, one operand can inhibit the other operands. And this is why we called it the excludon. And I put this slide because there has been just a paper published by a Spanish group, which has now designed a tool to really find this excludon, which I think are a new type of regulation in bacteria. Okay, so we did not find RNA which were involved in virulence. We found their regulation, and we found new type of regulation in small RNAs. I have put the word riboswitches. So riboswitches is a word which has been coined to talk about this region, which is upstream from an ORF, and which can, depending on the presence or the absence of a ligand, lead to the termination of transcription. This was in the case of ORF, open reading frame. We have found that this situation, this type of riboswitch, can also occur in the case of non-coding RNA. And we found two of these. which were regulated by B12. And I don't have time to tell you, but they seem to be very critical for the persistence of Listeria into the gut. So together with Rotem, we also found that there were many riboswitches, including riboswitches which regulate the resistance of Listeria to antibiotics. And we found a new type of antibiotic resistance, which is due to the splitting of the ribosome, which is something which should be investigated in more details, I think. Okay, so transcriptome, we had the genome, the transcriptome. I would like to discuss in one slide the translatome. I use the word translatome because we were interested to really identify the protein, the N-terminus of the protein, which were made by the whole genome. And we found a series of small open reading frames, and this one, PRLE42, was investigated in more details because we found it in many firmicutes, and we found that this was the link between a stress and a series of signaling pathways that you have inside the bacterium. And we also performed the structure of this protein. Okay, so I have told you about cellular microbiology approaches. I have told you about serendipity. I think the ACTE gene was really serendipitously found by my lab. I told you about postgenomic approaches. I would like to finish by hypothesis-driven approaches. I had the idea that similar to viruses, bacteria should modify histones when they infect cells. And Mélanie Hamon, when she arrived in the lab, started to investigate that. And it was obvious, but she found that indeed there are histone modifications which are induced by bacteria, but she found several modifications which were previously unknown, and she's continuing that lab now, her own lab now in the Pasteur Institute. Second, hypothesis-driven approaches, which was work by Fabricia Stravu. So this piece of work was done following a seminar that I gave, where I discovered that that mitochondria were not like beans on the left, but were very active and very mobile. They were able to fuse and fragment, etc. And I thought that probably Listeria was doing something on this mitochondrial network, or that mitochondria would affect the infection. So we started to investigate that, and we could show that indeed Listeria infection causes mitochondrial fragmentation. You will see what happens. You see, totally fragmented. And this happened very transiently. And LLO is inducing this transient fragmentation. And what is interesting is that contrary to the dogma, it is not DRP1, the dynamine-related protein, which is doing the job, but it is another component of the mitochondrial proteins, which is doing this fragmentation. So again, another window that we have opened by studying Listeria. Okay, and last, post-translational modification. We have studied two post-translational modifications which are ubiquitin-related, SUMOylation and ISG15. It's too bad because there was the picture of Liliana Rabosevich on the left, but in both cases, we found that the post-translational modification is critical for infection. So I think after immunology, which was really something very important in the field, using Listeria, I think we have shown that infection biology can be studied by using Listeria, cell biology, and epigenetics. We have found several new concepts in microbiology, in genomics and transcriptomics, starting with microbiology. So if you want to know more about all these, I think we have written several reviews that you can refer to. There are many things which are still to be done. I think at the level of the bacterium, at the level of the infection with cells, at the level of the whole infectious process, there are many issues that we have opened and are still to be done. I think it is important now to summarize. Listeria was discovered in 1926, not by Louis Pasteur or Robert Koch. It was discovered in England. I think immunology started in 1966 by the work of McAness. And then we started cellular microbiology in the late 80s, post-genomics after 2000, and epigenetics and infection and interaction with the microbiota is really in the air. So I wanted to say that if you look at this paper, which was published a few months ago, Listeria ranked 9. So it's one of the bacterium which has been highly investigated, and I think we contributed to that at least a little bit. And of course, I could not have done that without all this fantastic group of people. And with that, I would like to thank you very much.
Great. So many questions, but I'll just ask one. And that is dealing with your early work where you showed that E-cadherin is the receptor for the Listeria. And since E-cadherin is expressed in so many different cell types, is there something, is it just the fact that the Listeria is taken up by the gut through our foodstuffs that make it such an attractive site for the infection, or is there something beyond that that enables or facilitates the infection process?
Okay, first, I don't have the answer for that. The mice that we have generated are expressed under a promoter, which is specifically expressed in the gut. Okay, so you know now that the mice are not infected in the wild orally. So the other animals that we can use are guinea pig. So we have not investigating the infection along the whole animal in guinea pig for the moment. We had used other animals when we were studying the placenta, but I don't know. But I think it's a good thing. In fact, among the projects that are really prioritized now is to study the infection in vivo with all the tools that we have generated. I completely agree. But I don't have the answer for you, Hélène.
Thanks.
Very interesting talk. So intracellular bacteria have been found in many different systems. And is this open? So you talked about the gut, but now there's a lot of interest in intracellular bacteria in cancer cells. And and there is a lot of interest why would cancer cells have maintained intracellular bacteria. So I'm just wondering whether the mechanisms that you have identified, of course, these are different models and the bacteria may be different, but in terms of this signaling and interactions that could be, so in talking about it coherence also that have been present in cancer cells. So
Yeah.
I guess in your model in the end with bacteria in different systems, so I'm just talking about cancer.
Well, in the case of cancer, you said that there are many bacteria which are associated. I agree. I agree, there are more and more, but whether they are intracellular is another issue. For example, Bacterium nucleatum, or you have also Helicobacter pylori, which is inducing cancer and all that. So I don't think it is the intracellularity which is a key issue in the interaction between bacteria and cancer cells. In the case of Helicobacter, I think there is something which is very well known, that the inflammation and the constitutive inflammation is part of the cancer-inducing things. But also, from what I know, is that there are two issues. You have bacteria which are inducing the cancer, and also bacteria which like to be in the cancer cell. So I think it's two different issues, and I think there are many people who are interested in that, especially in the gastrointestinal tract. Mm.